Hello everyone. So in this lecture we'll discuss about physical mapping. We have already covered the basic tools and technologies which are involved in physical mapping of human genome. So in the physical map we proceed with cDNA and genomic DNA libraries. So cDNA can be ESTs and full length, and genomic DNA can be with large insert clones, STS markers, some of the markers which are polymorphic like RFLPs and all. And then we proceed with fish mapping and mapping with radiation hybrid cells, then contig assembly to come up with contiguous clones, and coming to finally to physical map. And sequencing is essentially involved in the physical mapping of the human genome. So this is a shotgun approach to sequence assembly. This is clone contig mapping. Again, by looking at the overlap, we tend to align the sequence. This is whole genome shotgun approach. So whole genome shotgun approach involves random fragmentation, size basis fractionation, then cloning in a suitable vector, then transformation, and then again inoculation, and then plasmid isolation sequencing. And after sequencing, contig assembly is done. So wherever the overlaps are there, the contiguous clones are formed, or clone contigs are created. This shows genomic DNA which transcribes to mRNA and then reverse transcribed to cDNA. Again this cDNA is cloned, then sequenced, and contig assembly is done. And finally the positions of these ESTs or cDNA basically indicates the position of the genes on the map. This is hierarchical shotgun approach, in which one basic back clone is utilized for fragmentation and the cloning purposes. So problems with shotgun approach are if there is wrong incorporation or wrong matching of the overlap. So here we can see this particular sequence is getting matched with this particular overlap. These sequences are skipped in between. So this becomes a wrong overlap. That is a problem with shotgun approach. Or if there are genome wide repeats, there is a problem in incorrect overlap. So sometime it may result in failure to it may result in errors when it is making the sequence of the chromosome. So wrong sequences can be linked together. So this is one of the disadvantage of the clone contig mapping. So the disadvantage is solved when we have some STS markers or some markers located on the clone or the insert which we are working on. So if we have few markers working on it, then we can have the correct alignment of these markers. So this misalignment of the sequences or misalignment of this kind of things can be avoided if we are using some markers. Now this is restriction library. So we know that restriction enzymes have specific site on the DNA resulting in the cut. Then RFLPs, they are used as DNA markers, and also restriction libraries can be prepared. So there are different fragments which are produced, and in this what actually is done, the reaction is done with two different restriction enzymes. So in one reaction one restriction enzyme is used, in another reaction another restriction enzyme is used, and in the third reaction both the restriction enzymes are used in a single reaction. So here we can see this is 4.9 kb fragment which is digested with EcoR1. So EcoR1 digestion gives 1.5 and 3.4 kb fragment. BAM-H1 gives 0.7, 1, 1.2 and 2 kb fragments. And when we are using both the enzymes collectively, it is giving 1.2 base kb sequence. So this 1.2 is present here, showing that this doesn't have any EcoR1 site in between. But here we see this is a 2 kb fragment, showing that this also doesn't have any EcoR1 site in between, and it is only cleaved by BAM-H1. Now this 0.7 is cleaved into 0.2 and 0.5 because this was cleaved by BAM-H1. So that shows that this 0.7 must be having a EcoR1 site in between, because in this reaction EcoR1 is present and it is able to give a cleavage in this giving cleavage in this pattern. And this 1 kb fragment is again present here, and these 1.5 we don't see in this one, and this 3.4 also we don't see in these two reactions. That means these fragments must be having BAM-H1 sites in between. So map 1 is created here. In map 1 the different fragments are marked like this. So and in map 2 the different fragments are marked like this. So if map 1 is correct, if we assume that the map 1 is correct, then the partial restriction products will include a fragment of 1.2 and 0.7, that is 1.9 kb, which is not present. And here if map 2 is correct, then the partial restriction produces will include a fragment of 2 and 0.7 which are actually present. So that means map 2 is correct. So this is how we can prepare a restriction map with assigning position of these restriction enzymes like this on the DNA molecule. Okay, and these sites can be polymorphic or they can be non-polymorphic. Now fingerprint mapping. In fingerprint mapping, the genome is broken into fragments which are then copied in bacterial cells. The DNA copies are then cut by restriction enzymes and the length of resulting fragments are estimated, and gel electrophoresis is used for separation of these fragments. So these are the fragments which are obtained, and when we tend to put them together, so these are showing overlap of DNA fragments using band pattern, then this comes a consensus restriction map. Now for Cdna library, the Cdna is prepared, mRNA has poly A tail, so we use poly DT primer. Poly DT primer, after using poly DT primer and reverse transcriptase enzyme, we have first strand synthesis by reverse transcriptase, and ribonuclease H is used to degrade most of the RNA after first strand is synthesized, and then second strand is synthesized by DNA polymerase 1 to have the double stranded cDNA. We can also proceed with single stranded cDNA for DNA microarray and other kind of work, and if we are using this for cloning purpose, then we will require double stranded cDNA. Now the ESTs are generated by sequencing cDNAs, which itself is synthesized from the mRNA molecule in the cell. The mRNA in a cell are copies of the genes that are being expressed. So this is basically expressed part of the genome. So if we map ESTs on the chromosomal map, that means we are coming up with gene maps. And mRNA does not contain sequences from the region between genes nor from the coding introns that are present within many interesting part of the genome. Now ESTs as genome landmarks, we can navigate the road maps to find destinations, like we open the Google map for destinations. We see that this is the road is going, there is a landmark which shows that okay, here we have a petrol pump, and after that we have to take a right turn, here is some site, here is some spot, the restaurant is coming. Likewise, we wish to navigate genes or important site on the genome map. So genome map will help us to navigate through nucleotides that make up our human genome. Now for example, if the arm of the chromosome is missing, so after the mapping part is over, we precisely know that what all genes were located on that arm and what were their functions. So what will be the outcome when the chromosomal arm is missing, or if we wish to locate what will be the outcome if this particular gene on this particular chromosome is defective or mutated, or the parents have the probability of having this defect and probability of passing it to their children. So once we have the genome map, once we have the marker assigned, we can easily navigate through the genome and we can easily type the function of the marker or the gene. So for this STS mapping was most powerful mapping, and in STS mapping the non-polymorphic markers which were widely used were ESTs. So sequence tag site is a short DNA sequence that is easily recognizable and occur only once in a genome. So its loci is fixed. So 3' EST, that is for 3' UTR, serves as a common source of STS due to their likelihood of being unique to that particular species and provide the additional feature of pointing directly to an expressed gene. So sequence tag site, it is used to generate detailed physical map of large genome. It leads to high resolution mapping, and restriction mapping and FISH, they are helping us, but neither of these two techniques will be able to help us for assigning more and more markers on the chromosome, because we have very few restriction sites on the total complete genomes, and there are very few ESTs also to be mapped. So to saturate the map, we need some unique locations which are which help us in navigation through the genome. So STS is one of the most powerful mapping tool which has helped us to come up with marker framework map. So sequence tag site or STS is simply a short DNA sequence, generally between 100 to 500 base pairs in length, that is easily recognizable and occurs only once in the chromosome. So the loci is fixed and it has got a fixed position, so it is very easy to map a set of STS from a collection of overlapping DNA from a single chromosome or from the entire genome. So, from a single chromosome, we have already discussed the flow cytometric method for isolation of single chromosomes. An entire genome can be done by whole genome shotgun approach or restriction library. So, here these STS will help us in avoiding any kind of wrong information or wrong overlapping between the contigs, the overlapping DNA fragments, to come up with the correct DNA fragments. So, this is how we can see these are pairs of closely linked marker. These are pairs of less closely linked marker. So, as you can see here, when the fragmentation is done, so these two markers, since they are very close to each other, so they tend to be together when the fragmentation is done. So, this was the basic concept of radiation hybrid mapping, in which STS marker mappings were done, and it was assumed that if the marker is having distance between them, then there is a probability that they can be separated by fragmentation. So, here we can see this is separated, here this is separated, this in this one it is retained, but otherwise it is separated in most of the fragment collections. So, the more closely markers are linked, the more closely markers are located, the more chances are that they will be retained on the same fragment during fragmentation pattern. So, the data can be helpful to calculate the distance between two markers in a manner which is analogous to the way in which map distances are determined by linkage analysis. So, this has largely helped in the genetic mapping of the human genome, because in linkage analysis a map distance is calculated from the frequency at which crossover occurs between the two markers. So, STS mapping is essentially the same, except that each map distance is based on the frequency at which breaks occur between these markers. You can see the chromosomal fragmentation pattern. So, how if they are close, the markers are very close to each other, there is very rare possibility that the break will separate them. So, this is a basic principle of the linkage mapping of using STS markers, and this has helped us to come up with marker framework map. So, STS mapping, this maps the position of short DNA sequences between 200 to 500 base pairs in length, and they are very unique. They have unique location on the chromosome, and to map a set of STS, a collection of overlapping DNA fragments from single chromosome or the entire genome is required, and genome is first broken into fragments, then it is cloned in bacteria or some other vector, and then it is transformed, and then the sequence is sequenced, process for sequencing, and then it is looked for overlapping DNA sequences. Now, here we see there are some problems associated with YACs, because we discussed initially that YACs are prone for rearrangements. So, here we are trying to map some STS on the East artificial chromosome. So, these are panel of STS which we are trying to map on East artificial chromosome. So, these are some representation of East artificial chromosome, and we can see that there are some gaps. So, these gaps are present in East artificial chromosome for these particular markers, and these are because of the rearrangements which have occurred in East artificial chromosome. So, because of these problems, the more reliable vectors, because YACs were preferred for their high capacity, but reliable vectors were bacterial artificial chromosome, and they were also mapped with radiation hybrid panel of radiation hybrids for coming up to final data. So, this is again process of STS mapping is shown. One by one STS are mapped for overlapping DNA fragments to generate clone contig mapping. Again, in this different clones, these are random fragmentation, and these in this we can see that there are a particular STS which are maintained in particular clones, and then clone library is used as mapping reagent, and these STS are mapped for their original aligned sequence, for their original alignment on the chromosome. The same panel of STS, different STS are typed on panel of radiation hybrid cells. So, this was very important advantage of STS mapping, that the individual clones were sequenced, subsequently they were able to provide the DNA which is actually sequenced, and then the clone contigs were assembled, and continuous DNA sequences were generated. So, this STS was very helpful to come up with marker framework map. So, in clone contig approach we have discussed the genome is broken, it is sequenced, and the overlaps are basically aligned. Fluorescence in situ hybridization, we have discussed, it was done to know the position of the different probes or markers. Let's say we have come up with clone, contiguous clone, and we wish to know to which chromosome it belongs to. So chromosomal in situ hybridization is a method of choice which helps us to localize the position of the probe on the chromosome. So likewise, after we assign the position of these on the chromosome, then we know that okay, this particular clone or this particular radiation hybrid or this particular sequence belongs to which chromosome. Now we see all the markers together. So this is human chromosome. In the red we see that this is comprehensive restriction radiation hybrid mapping. So radiation hybrid mapping has provided us with chromosome framework map. Then high confidence bin lines, they are shown in blue, and genetic markers are shown in the green one, and the express sequences, which will be basically gene markers, they are shown in these purple colors. So you see that some of the chromosomes will have large number of ESTs, but some of them will have less number of ESTs, showing that some of the chromosomes are gene rich and some of the chromosomes are gene poor. So this is how radiation hybrids, they have helped us to create marker framework map. Now again we'll go to the basic again we'll go to the basic chart of the DNA mapping, in which physical mapping was basically initiated with cDNA and genomic DNA clones. So cDNA clones can be EST full length, genomic DNA can be large insert, can be done with STS. Finally, to know the chromosomal localization, we have used fish mapping and mapping with hybrid cells. Then contig assembly was done to create contiguous clone. Sequencing was essentially involved to come up with physical map. So this part will basically cover the physical mapping of the human genome, and physical mapping also go in hand through hand with genetic mapping, because EST, when we are trying to saturate EST on the chromosomal map, we are basically trying to come up with the gene maps. Basically here also, when we are doing disease inheritance and looking for the mutations and all in the pedigree analysis, that is also done to come up with what genes are linked, what kind of markers are linked with the diseases. So this some part of physical map will help us in unraveling the genetic map of the human genome, and all data combined will give us human genome map. And these are some of the selected readings for this part of the mapping, that is physical mapping.